So we'll begin now. Hi everyone and welcome back to session 2 of the Epigenetics course. And I hope you had a good time in the last session. We will start today by discussing the assignment which is provided in the last session. I would also like to remind you to turn in the assignments by tonight and to use the chat box if you have any questions. So let's dive right into it. So if you remember the assignment, it had a question to decode a code and most of you got it right. It said, I like genetics, and which is why you're here. So the first question asks us, write the three characteristics of the genetic code. We will start by stating three basic answers to that. First, one codon is made up of three bases or is in a triplet. Second, some of you responded that the genetic code is universal, but do you actually know why? It can be explained through an example. If you isolate the gene that is responsible for making insulin in humans and you replace it with a bacteria, the body will still produce insulin since the genetic code is universal. That explains the second answer, that the genetic code is universal. Third answer to this is, in the human body, there are 20 amino acids, excluding the ones which are not widely recognized. But still, you have 64 combinations. Again, can you guess why? The answer is that the genetic code degenerates, meaning although one codon is specific for one amino acid and is responsible for coding one amino acid can also be coded by one or more codon. The second question asks, during the class, you learned about central dogma, DNA to RNA to protein. Can we go reverse, RNA to DNA? As explained in the previous session, the restaurant analogy by Ishani, have you ever thought if it could be reversed? And the answer to that is yes, it can be reversed, and RNA could be reversed to DNA by the process of reverse transcription. It is common in viruses and is seen in vaccines, and will also be explained in the upcoming sessions. Over to Sandhya.
Yeah, hi everybody, good evening. Okay, so this class is going to be covering what epigenetics is in the mechanisms of it, if in how it very physically how it works. So it might get a little heavy, so feel free to drop any questions that you have in the chat box, and I will be pausing occasionally to take any questions that you might ask. So please feel free. So I'm going to start by calling back a slide that we saw in session 0, if you guys have seen it. So we know that you have a zygote. We all become who we are from a single cell zygote. And since all the different cells in our body originate from that one single cell, they do have the same genetic sequence. All the genetic sequence in all our cells is identical. But then we also very clearly know that they're so different, right? Your hands, you can feel things with your hands. You can't feel things in your internal organs. You don't feel your heart beating every time it beats. You don't feel food going into your stomach. So you have so many different types of cells, and they have different functions, they have different appearances and characteristics. So how does this happen if they all have the same genetic sequence? Considering that the course is called epigenetics and you guys have attended the previous session, you know that the answer to this is epigenetics. But let's dive into the actual details of it. So think of epigenetics as a sentence in English where you have a fundamental primary sentence. So that's your genetic sequence. And the meaning that you derive from that sentence is different based on where you apply emphasis, what parts of it you choose to read. So it's the same thing. Look at this sentence. I did not eat 10 cupcakes. You can say, I didn't eat 10 cupcakes. Maybe my friend did. Or, I did not eat 10 cupcakes. I only baked 10 cupcakes. Or, I didn't eat 10 cupcakes. I ate 10 donuts. So the emphasis, where you apply the emphasis in the sentence, changes the meaning of the sentence. So your primary sentence is not changing, just the things that you apply on that primary sentence change. So your genetic sequence is not changing, but what parts of your genetic sequence you decide to give emphasis to, what you choose to convert from genes to mRNA to protein that we've studied changes what the cell looks like. So we know that anything that's in a cell is because of the proteins that are in it, right? So we know that the DNA becomes RNA, becomes protein, and that's the central dogma, which we've studied in session 0 and again last week. And so we know this. So if we have to figure out how epigenetics actually works, these are important things to keep in mind. Another thing that we know is how DNA exists. DNA is coiled around protein structures, and we've covered this. So who can tell me what these proteins are called? So these proteins are called histones, and they form a nucleosome. So these this is how DNA exists. So now if I have to change how my how my DNA is read or change what parts of my DNA is read, what can I modify? I can modify pretty much any of these steps. So, and keeping in mind that my final aim is to modify what proteins are present in my cell. So this is a short video that talks about what epigenetics is. Sorry, 1 second. I'm sorry about that. Um, is it audible now?
Still listening.
Sorry.
Not audible.
Sandhya, you may have to change your sharing settings. Advanced settings in sharing.
Oh yeah, I tried doing that. That's what I changed.
Do you want me to try presenting?
No, it's okay. Okay.
Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein.
So that's basically what epigenetic tags are about. So we will get into the details. So the first thing that you can change are histones. So we know that DNA is wrapped around histone proteins, and these proteins have tails, like you can see here, on which different chemical groups can be added to modify it. So the histone is positively charged and your DNA is negatively charged. Now, that's why they interact. That's why your DNA can bind around the histone. So, the basis of any change that happens to your histone is to alter that charge. So, if I make my histone a little more positive, it'll bind a little better to my DNA, and so on. So, that's the idea behind any change that happens on your histone. So, the first is methylation. A methyl group, as a lot of you will know, is basically just CH3. So it is a methyl group can be added to your histone, and a methyl group is positive. So your histone is positive and your methyl group is positive, which makes the complex doubly positive. So this means that your DNA will bind better to it, and that forms slightly compact chromatin. And this, as we've learned before, is called heterochromatin. So when it's so tightly bound, the enzymes that need to access your DNA in order to convert it to mRNA and then to proteins cannot access it anymore. So your genes are silenced in the sense that the proteins that they code for are not actually made. So that's histone methylation, and this is carried out by an enzyme which is called histone methyltransferase. So transferase means it has to transfer from one thing to another. So it transfers your methyl group from SAM, which is a very common chemical in all your cells. It transfers your methyl group from SAM and puts it onto the histone, giving you SAH. So if you take out the methyl from SAM, you get SAH. The next modification is histone acetylation, and this is the exact opposite of histone methylation. Acetyl, as you might have heard in acetic acid, right? It is negatively charged. So an acetyl group is a negatively charged group. And when when acetyl is added to your histone, your positively charged histone becomes less positively charged. So if it's less positively charged, it binds less tightly to your DNA. So you have loose chromatin, you have more open chromatin. And if you just look at it, you can see that there's space, right? So there's space for enzymes to come and attach to your DNA so that they can convert that to mRNA and convert that to proteins. So in any specific cell, if I want the gene to be expressed, what I might do is I might add a acetyl to my histone so that the DNA is accessible so genes can be expressed. If I want to silence a gene, I might add histone. I might add methyl to my histone so it becomes tightly bound so I cannot express the gene. And this process is mediated by histone acetyl transferases, which transfer the acetyl group from acetyl coenzyme A. And so the acetyl goes from here onto your histone, giving you just coenzyme A. So just like most of these changes that happen in your epigenome or all your epigenetic changes are reversible. So if they can be added, they have to be removed as well. Because as we know, your epigenome can be changed by the environment that you live in. So if I have to be able to respond to the environment, then I have to be able to change, right? So that means it has to be easy to write and it has to be easy to erase. So each epigenetic modification has writers, erasers, and readers. Writers write, so enzymes or proteins that write onto another protein are called writers. Erasers are what undo this process, and readers recognize this process. Because what's the point of acetylation or methylation if it is not recognized by something? So the enzymes responsible for this are histone demethylases. So methyl, demethyl. Demethyl is removing the methyl, and histone deacetylases, also called HDAC. So next comes the one of the most important epigenetic changes, and this is DNA methylation. So here methylation happens, and the process is the same, but it happens to DNA. So it happens to your cytosine base specifically. We know that DNA has A, T, G, and C. So a methyl group is added again from SAM. It's added onto your cytosine here. As you can see, this is your methylated cytosine. Now, this is what DNA methylation is, and we'll get to the function a little. But so scientists studied a lot of people's genomes and tried to identify methylated sites. And and as you know, we can sequence our genome. So what they realized was that a lot of the cytosines that were being methylated came in a dinucleotide or a pair. So when you have C and G together, it's very likely to be methylated. So why? Why is that? Is it that C and G, when they're next to each other, the DNA is a certain shape? No, it's not. The reason is actually very simple. What it is that whatever change you have on one strand of your DNA should ideally be reflected on your other strand of DNA. So if I have C and G right next to each other, as you can see, this methyl group here is reflected here. And it is reflected only because a C pairs to a G and the G is right next to this C. If, say, for instance, this G was a T, right? This would be an A, and you wouldn't have a methyl group because A does not get methylated, C gets methylated. So, for the simple reason that whatever change you have on one strand of DNA needs to be on the other strand of DNA, it's CG that gets methylated. And this is important because any change that happens in epigenetic modifications in one cell of your body, what is the point? What is the point of that change if it cannot be propagated to the other cells of your body? It has to be propagated every time your cells split or every time your DNA multiplies, which is mainly during cell division. Every time your DNA multiplies, that change has to be carried forward, right? And as we will see a little later in this presentation, that this carry forward is not just in one cell of your body to the other cells that come from it, but from parent to child, across generations and across organisms, these epigenetic changes are carried forward. So we understood why histone modifications take place because they facilitate either DNA binding more tightly or less tightly around histone proteins. But why should we methylate DNA? So some of you might know this, but so when you have a gene, so look at this thing on the left, for instance, the bright blue is your gene. There is a sequence before your gene, which is called your promoter sequence. It promotes transcription of your gene so that your gene can become mRNA, can become protein in the whole process. So and this is mediated by an enzyme which is called a polymerase. So your polymerase, how it does is it comes and it binds to this promoter sequence. So your enzyme binds directly to your promoter sequence. This ATCG that you can see here is your promoter sequence. So when your polymerase binds to your promoter sequence, gene expression takes place. It's turned on. But look at this case when it's methylated. So you have CG duplets here and all of them are methylated. When this happens, because of pure physical reasons, your polymerase cannot bind to your DNA because your methyl group is already bound to it, right? So there's no way your polymerase can interact with your promoter. And if your polymerase cannot interact with your promoter, transcription cannot take place. So your gene is turned off. So this is how DNA methylation basically contributes to different cells expressing different genes. So in my heart cell, one set of genes will be turned on. in my lung cells, another set of genes will be turned on, and this is mainly mediated through histone acetylation, methylation, and DNA methylation. This, the genes which produce proteins that are important for the functioning of that particular cell are turned on. So, like in my stomach, any genes that code for, say, enzymes, that digestive enzymes, so genes that code for digestive enzymes will be turned on, but other genes that I don't really require, like for sensitivity or something like that, might not be turned on. Those might be turned off. And it is this combination of what genes do I turn on, what genes do I turn off in the different cells in your body that make them all unique, that make all your cells different. So another thing I wanted to mention is, because since CG is where most methylation takes place, and this has best impact when it's in your promoter, right? Because if I have this really far off from this gene, it's not going to directly be able to impact how this gene is expressed. So there are regions in your genome called CPG islands. So think of your genome is an ocean, and there are some parts which are promoters of genes where CG duplets are very, very common. So these are called CPG islands. And this is where DNA methylation usually takes place. So these are really cute animations that tell you what exactly DNA methylation does. So the methyl group on your DNA says stop to your transcription machinery that now cannot bind to your DNA anymore. And these are DNMTs, which is DNA methyltransferases, that are adding methyl groups onto your DNA to prevent uh, gene transcription. So, just for a quick question, there's no way too many of you would know this as such, but what percentage of our CPG islands in our genome do you think are methylated? That's basically saying, if I have 100 genes in my genome, how many of them do you think are actually active? So there's a link in the chat box that you can access and respond to the poll. That's very interesting. The fact that that average is just above 50%, that's 60%. We have three groups. We have one group of people who think it's really, really low, and we have couple in between. So, anyone else need to vote? Okay, so the answer is about 80 to 90%. Because imagine if your one cell has the genes that can make every protein that your body is ever going to need. You're never going to have a cell that requires too much of it, right? So 80 to 90% of your CPG islands are methylated on average. So I'm going to pause here for a little bit, and do any of you have any questions? Do you have anything that you'd like me to explain again if you haven't understood? Anything that wasn't clear? Please feel free to unmute and ask. Okay, just a heads up, we have a quiz at the end of this presentation. So please do ask any questions that you might have. It's a pretty simple, straightforward quiz if you've understood everything. Okay, I'm going to take that as I did a great job and everyone understood. Okay, next. Moving on. So, like I said, most epigenetic modifications that happen are reversible. So, like your histone modifications were reversible, your DNA methylation modifications are also reversible. But there's a catch here. The catch is that addition of a methyl group to a cytosine gives you this bond right here, which is a C-C bond. And C-C bonds are very hard to break. So there is no chemical that can just come and directly just cleave that bond as very simply. So what you have is it's a longish process that consists of two steps. So say you have methylated to methylated cytosines here. The first process that happens is called deamination, which is removal of an amino group. So amino group, as some of you might know, is an NH2. So your NH2 group is removed, and this after deamination gives you thymine. So methylated cytosine, if you remove the amino group, gives you thymine. And thymine is also one of the bases of your DNA. So you have a methylated cytosine here. After deamination, this C gets converted to a T. But now you all know by now that an A pairs to a T and a C pairs to a G, right? So when your cell sees that there's a T and a G here, it's flagged as a mismatch. The cell realizes that, hey, look, this pairing is wrong. This is not how it's supposed to be. And so it starts the process of DNA repair. That is a whole other process, which I'm not going to get into. But ultimately, the aim is to correct that mistake. But the cell doesn't know which the original base was. It doesn't know if G is the mistake or T is the mistake, right? So there's equal likelihood that any of them will happen. So it might say, okay, T is the mistake, make that a C so that the pairing is corrected, and you will get this, which is the same as your initial sequence, so all's good. But then you might also end up changing a G. And if you end up changing a G, you get an A here. And these two sequences are not the same. So then you have mutations that might arise because of DNA repair. So that's the risk with too much methylation, that you might have some mutations that get accumulated. So to sum it up, histone acetylation forms euchromatin, and euchromatin is loosely bound. So histone acetylation forms euchromatin. Its writers are histone acetyltransferases, and erasers are histone deacetylases. And histone methylation forms heterochromatin because it does the exact opposite of what histone acetylation does. Its writers are methyltransferases and erasers are demethylases. And here, if you're wondering, the K is the residue on which your histone gets methylated, called a lysine residue. So we studied in the last class about the genetic code and your amino acids. So every amino acid has a single letter code. So the single letter code for lysine is K. So lysine demethylases. Okay, so then DNA methylation inhibits gene expression, written by DNA methyltransferases, and erasers is basically through deamination. So this image here looks a little complicated, but in essence, what it's actually saying is that no epigenetic modification acts alone. There is always interaction. So if histone acetylation takes place, it is very likely going to be accompanied by histone demethylation because that only makes sense. If I add a negative group to my histone, I might as well remove the positive groups because that just makes my process more efficient, right? And so that's exactly what this is doing. So if you look at these small blue dots, those are DNA methylations. That's at CPG sites, the C's are methylated. So DNA methylation is bound in through a process, through a series of interactions. In it calls upon HMT. HMT is histone methyltransferases. So by methylating your DNA, you're also going to methylate your histone. And as you can see here, you have a HDAC. A HDAC is a histone deacetylase. So like I said, you have histone methylation and histone deacetylation that happen together. Yeah. So this is a small simulation of this whole thing. You've seen this simulation last session. And this time you can see it with the different epigenetic modifications that might be there. Okay, that's taking a while to load. So moving on. So you have, so after tags that can be added onto your DNA, like histone acetylation, methylation tags, you have another process called RNA-based regulation. So your genetic sequence or your gene expression can be regulated by RNA molecules. So let's find out how that happens. So we know that there are three basic types of RNA molecules, right? We've learned that last class: rRNA, mRNA, and tRNA, and they all have their own functions. But there's another kind of RNA which is called non-coding RNA. And these, as you can say, they don't code for any protein. So what is non-coding RNA? So you have this top one here is your DNA sequence. After transcription, you get mRNA. And in case of non-coding RNA, after transcription, you get non-coding RNA. In case of mRNA, that moves on to form a protein, because the point is your mRNA codes for a protein. And this process, as we've seen before, is called translation. But in case of non-coding RNA, there is no translation. Translation does not take place. The functions that non-coding RNA carry out are carried out in this form. They don't form a protein that carries out functions. This non-coding RNA interacts with different components to give results. So what does it do? So the first thing is non-coding RNA can inhibit transcription. So by inhibit transcription, it causes chromatin modifications, or it interferes with transcription. So it binds to your DNA. So it'll your one when your DNA needs to be transcribed, it opens up. And when it opens up, this non-coding RNA can go bind to it. So then transcription cannot take place. Your enzyme that forms mRNA cannot glide along your DNA if it has formed a duplex. And second, it can also inhibit translation. It binds to your mRNA. You have a single stranded mRNA and you have a single stranded non-coding RNA. If they have any sequence similarity, they will bind together and they will form a duplex. And this duplex cannot do anything. So RNA interference is a technology that was developed based on this. So like what I said, so you have mRNA, which is in red, and you have non-coding RNA, which is this purple, and the non-coding RNA binds to the mRNA. So they form a duplex, which is bound together. That's a hybrid molecule, right? No, that's two different kinds of RNA together. So this forms a duplex, and because it's two strands, it cannot be read by a ribosome, and so it cannot form proteins. Ribosomes, as you will remember, are the organelles that are responsible for converting or forming proteins based on a mRNA template. So if it's two strands, it cannot interact with your ribosome, and so you don't get protein. But the inhibition doesn't stop there. What happens next is that this duplex molecule is cut down into many small fragments. And with a very small fragment, it has no function in the cell, so it's recycled and stuff like that. But the point is that a target mRNA so you can target an mRNA for degradation, for complete degradation, by creating a non-coding RNA that binds to it, that's complementary to it. That's what RNA interference is. You have an RNA molecule, which is your non-coding RNA, that interferes with the translation process by binding to mRNA. Make sense? So RNA interference has a lot of different applications. One main application is in research. So if I want to find out a function of one gene, I have gene A and I want to find out what it does in the cell. The easiest way for me to do that is to silence gene A and see what happens to the cell. If I see that the cell becomes a different shape or it has no clear shape now because I've silenced A, that means that A was responsible for maintaining the shape of my cell, right? So that's one application of RNAi. It's also been explored that RNAi might find applications in therapy where it can be targeted so that you can target proteins that do you harm, that are pathogenic. So proteins from pathogens, from viruses or bacteria that might infect you. So I'm done with that. So are there any questions or doubts? Anything that I can clarify?
Sandhya, there's one question here by Premlata Chauhan. Is turning on and off of the genes by methylation related to switching on or off of operon?
Or no. So in an operon concept, methylation does not really exist, at least in the well-studied ones. But it's the same. It's a similar kind of function. In an operon, repressor proteins can bind to. So it's a similar function. So in an operon, other proteins bind to your promoter so that transcription cannot happen. But in methylation, methyl groups bind to your promoter. So it's very similar, but in an operon, usually methylation doesn't come into the picture. So if that's all, then I hand it over to Arushi, who will be talking to you about why we need to study epigenetics.
Yeah. So hi everyone. Epigenetics, as we know, the science of change, is a rapidly growing field blazing with great opportunities to provide in the future and even now, and explore the world as well as various different fields from a different perspective. Before I delve deeper into this, I would like to ask you all, why do you think epigenetics is something one should study? Why is it significant? You can either unmute or say or write it in the chat box if you guys have any answers.
To understand changes that environment has on us, like why we are the way we are.
Yeah, that's a great point. Glad to hear your thoughts on the significance of epigenetics. Um, yes, absolutely, Avril, you're right about that. So those were some great points. Now I'll move ahead. Can we move on to the next slide? Thank you. Epigenetics is not limited by boundaries. There are tons of areas to investigate that still haven't been thoroughly touched upon, and enhancement or upliftment of various other fields. Here are a few of them. Better understanding of epigenetically inherited diseases or its components in other diseases like memory, cancer, autoimmune disease, and even addiction. Epigenetics and medicine, epigenetic therapy, epigenetics in agriculture, epigenetics, stem cell differentiation and development, epigenetics and psychology is an extremely intriguing pair, and we'll look more into it in a few. And lastly, we have nutritional epigenetics. Next slide. Now let's check some of these out in a slight detail. All right, so we have epigenetic therapy. It is to treat medical conditions like cancer, heart diseases, or diabetes, and several others that have roots or are influenced by epigenetics through drugs and other epigenomic like techniques. It has shown some promising result in cancer treatment when given along with chemotherapy as well as radiation. And well, epigenetic therapy is a lot based on epigenetic drugs. So, considering that a number of epigenetic factors are reversible with a combination of epigenetic therapy and epigenetic drugs, which basically work on the concepts or are based on the fact that most of these changes that we have looked at right now um, are reversible. And DNA methylation, histone methylation, and acetylation can alter gene expression. These drugs target these modifications to alter the gene expression. Next slide. Then we have stem cells. Stem cells, there have been increasing evidences that suggest that epigenetics play a significant role in stem cell biology. They are important for determining gene expression during cell differentiation and development. Next, we have agriculture. Through epigenetic control mechanisms, crops are altered in such a way that they gain the ability to respond to our constantly changing environment, the environment and challenges, and the stress during the growth and development of a crop. It is a great application of epigenetics. This could lead to a future with climate smart crops. Next slide. Next. Yeah, thank you. Um, Now we have psychology and epigenetics, and the implications of epigenetics in the field of psychology. Epigenetics and psychology helps us to understand how the expressions of gene is influenced by expression, nature, and nurture around an individual to produce an individual with differences in behavior, cognition, cognitive abilities, and even personality, as well as mental health. There are certain epigenetic mechanisms and psychological disorders that are also included in this field. Let's look at the picture of the kid here holding a teddy bear and discuss how early life experiences exert a long lasting influence on physical as well as mental health throughout life. Having a tumulus family since birth can increase one's likelihood of schizophrenia, which is an epigenetically inherited disease, and can also have strenuous effects on the individual's mental health. Sorry. But how can something happening in one's environment affect their behaviors, their mental health? I mean, if you look at it in a way, everyone has the same set of um, genetic material. So how is it that people are so different? Well, this is what isn't this what epigenetics is all about? Particularly in this study, learning and memory, emotion, and social behavior in humans is understanding the initiation and maintenance of epigenetic states, and thus is an important aspect of research in the current field of biology. It is an intensely fun and a rapidly growing area, attracting a lot of attention. So, if you are interested in any of these, please take out some of your time and check them out. Thank you. Over to Kartavya to take the session ahead.
Thank you, Arushi, for a wonderful session. So, by now, you might be familiar with all these examples, the rooty mice, twin sisters, and calico cat. Today, we shall be diving deeper into the epigenetic studies of calico cat. So, why do you think the typical phenotype of three coat colors, the white, brown, and black, would be present in calico cat? Can anybody have give a wild guess? Okay, it's due to X inactivation. The phenotype you observe is due to X inactivation. So, what is this X inactivation? X inactivation is a mechanism by which one of the two copies of chromosome X in female mammals or in marsupials get inactivated. This happens generally randomly, but it has a very complicated process in back end. We can either get a paternally inactivated X chromosome or maternally inactivated chromosome. But how does this happen? The process is quite complicated, but I will try to simplify it. Uh, in the diagram, if you see, there is region, blue region marked in on the A figure A, that is axis. It's found in the region called XIC, which is X chromosome inactivation center. As the name suggests, this center is responsible for controlling X inactivation. The gene exists, which stands for X chromosome inactive specific transcript, uh, is transcribed from the gene, and which later covers the non-coding RNA produced later covers entire X chromosome, which is destined to get inactivated. But it's not always that this choice is made. Uh, Either of depending upon the uh, on the availability of transcript and the time pertinence, either of the X chromosomes gets inactivated. But initially, both X chromosomes transcribe this gene. Once this gene has been transcribed and RNA has coated entire X chromosome, it gets this gene gets silenced and no other protein which are required for transcription or translation gets attached to the chromosome, and therefore it's regulated. But another thing which regulates this process is called TRC complex, which is a little bit uh, which has a little bit 2 to 3 genes, which altogether facilitates this environment. Uh, but once this coating has been done, then but but if you think not everything is permanent. RNA, non-coding RNA has a limited lifespan. Then, if you think once their lifespan has ended, they should disappear or chromosome should get reactivated. But it is not the case. Once RNA has covered the chromosome, DNA methylation and X chromosome and histone modification occurs, like observed in the triangular print on the gene along in the XY chromosome. This helps in further silencing the chromosome. Now let's move on further. Uh, this uh, this type of if you can see the image, if in this is how the calico cats are formed. In one cell, suppose yellow chromosome gets inactivated, so the phenotype will get black uh, cell which will encode for black color. Another yellow cell will be formed, and once these divide, uh, we would see the peculiar two-coat color of calico cat. If you can see the diagram here, it it shows how an inactivated chromosome X and activated chromosome look like. The red dots where here you see means the RNA transcript which causes X chromosome to get inactivated. While since there is no RNA transcript for X chromosome, it would remain free and it would express the gene which is intended to be expressed. Similarly, this diagram also shows the inactivated region, inactivated X chromosome. Uh, Now let's watch a quick video so that you can uh, you can find it more interesting.
And these cells start off in a female with two X chromosomes. The cell divides, and the embryo is getting slightly bigger, and still each cell has both X chromosomes.
Are you able to hear the sound?
Yes.
Yeah.
Active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X as active, while the other one, the maternal one. And now this process will happen at random, and you'll have almost half of the cells with the maternal X active, half with the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat color gene is on the X chromosome, and that's precisely why these cats look like this, a mosaic appearance, because of the process of X chromosome inactivation.
Okay, before
And these cells start off in a female.
before we move on further, if there are any questions, you can ask now. Any questions until now? I think no. Then next top.
So we have a question here. Are all spots, markings in cats or other animal caused by this reason? I mean, X chromosome inactivation.
Yeah, most of the spots which are observed in the animals belong to the female animal, and that's the reason. Because if we see all the males inherit to one X chromosome and one Y chromosome. In since this X inactivation occurs in uh, since there are only one X chromosome, it would work normally and it would express itself normally. So we would be seeing just a single color like black, white, or any other color which is dominant trait. But in females, since there are two X chromosomes, and if one X chromosome gets inactivated in one cell, one other X chromosome would express itself, and we would then see the patches like we see in some animals. Uh, did you get your answer? Okay. So the next topic I would be discussing is genome imprinting. Uh, until now, you have heard that we carry two alleles. We are biallelic, and both of these are expressed, if not in the same time, but in different time, because one chromosome is because uh, if not in one time, but in different timelines, if, uh, both alleles are expressed. But in case of imprinting, only one copy of gene gets expressible. This phenomenon is called imprinting of genes. The major mechanism by which this occurs is promoter methylation. Since we haven't done this, but if you can, if you could imagine the broad line, the gene can be divided into three parts. First is enhancer region, second is promoter region, and third is the gene itself. While enhancer helps in regulation of gene, promoter defines uh, how the gene will be activated. If your promoter gets silenced or gets methylated, then the gene won't be expressing itself. And this is what happens during genomic imprinting. When the promoter gets silenced, and therefore there is no gene activity. So now, can anyone tell who are these two animals? A guess would be right. Uh, if someone can do.
Is that a liger, like a crossbreed between tigers and lions?
Yeah, the left one is liger, a female liger, and another one is tiglon, male tiglon. And these both, uh, if you see, both are derived from tigers and lions. The gene imprinting happens in these types of animals, which causes the difference in size and appearance. Like if you compare these two with the original self, like a lion or a tiger, you would see that they look very different from both of that animal, both of those animals, and we get uh, that's why gene imprinting defines the size and appearance of the animal. To take a few examples of gene imprinting, our gene has more than 100 imprinted genes which have been experimentally identified, but still there are several others which are being experimentally derived, but they have been theoretically predicted but haven't been identified successfully through experiments. A few examples you can find here is IGF2, which is which is insulin growth factor 2, H1, H19, and NDN. IGF, take note of IGF2 because we would be discussing it further in our next section. IGF2 is a paternally active gene. That means it's expressed by paternal exosome, and in maternal exosome it is imprinted or it is silent. If anybody wants to have a look on other genes present in humans in imprinted form, they can go on to this catalog of parents of origin effect, which is a database primarily developed for imprinted genes. But about the genomic imprinting, we shall be discussing it in further sessions for more information. Now let's have another quick video so that you can understand.
So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, clone probably has a tattoo on its face. And if clone had stayed in the circus, clone probably, professionally rides a unicycle over a tightrope, so clone is probably in pretty good shape. But then again, clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that to do start growing. But even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know, it's such an unbelievable buzzkill. There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors a parent experiences can be passed down from generation to generation was sparked in the 1980s. And this happened when some scientists were looking at the birth and death records of some people who lived in 19th century Sweden. This is a weird place to find a genetic revolution, but there it is. It wasn't just any place in Sweden. It was in Norrbotten, which is the northernmost county in Sweden, which is literally in the Arctic Circle. And despite the fact that Norrbotten was literally the worst place you could possibly choose to live in Sweden, there were some people living there in the 1800s. And they were completely cut off from the rest of the world. Let me clarify. These people were isolated. Like, if they didn't have a good crop year, people died. No, it wasn't ideal, but, you know, they didn't ask me my opinion, so I'm not giving it. So anyway, these people subsisted entirely on what they grew and the animals that they raised. And like I said, sometimes they starved. But sometimes they had huge, bountiful years of plenty. And when it happened, people totally went ape crackers. I mean, of course they did, because they were so freaking hungry, and then all of a sudden there was all this damn food everywhere. Anyway, there was a public health specialist who was looking at the effects of the people who grew up in the really bad starving periods of time versus the people who grew up in the eat all you can at the smorgasbord years. And you might already be guessing what they found out. People who went from relatively slim pickings to feed your face until you have to barf and then do it all over again in a single season, those people died an average of 6 years sooner than their starved out counterparts. And you know what sucks? So do their kids. And so do their kids' kids. Boo epigenetics. So, of course, now we all know, and we're all going to stop doing unhealthy things starting today. Unless the damage is already done. The damage is almost certainly already done. But hey, epigenetics brings good tidings along with the bad ones. For instance, we now know that certain types of cancer are caused by misplaced epigenetic tags, and scientists are now developing drugs that can silence the bad genes that were supposed to be turned off in the first place. Additionally, until recently, we thought that genes were the end-all and be-all of who you got to be. They were your blueprint, and you couldn't escape them. This outlook is not just kind of depressing, it also leads to a yucky sort of social prejudice. Because when you look at data without considering social and epigenetic factors, it might look like people with less money are less intelligent. So just like 15 years ago, there were scientists saying things in public, mind you, like, some people just have good genes for intelligence, and it just so happens that the poor people don't. Oh, it's so sad. Too bad, poor people. But you have bad genes. Well, it turns out that that is not even a little bit true. Not only are there a huge variety of social factors that affect how well people do on intelligence tests, but a genetic trait is also not just a product of genes, it's also a product of environment. Any one person's genome is determined by any number of decisions made by any number of their ancestors. And right now, you are making decisions that are going to affect people who are alive long after you're dead. No pressure or anything. And so I'm glad you've watched this, because when you and your clone are in your 60s and you sit down, have lunch, and he brings his son and his son has a tattoo on his face, he did not inherit that from you. This is Hank Green for The SciShow. We hope you learned something.
The whole point of showing this video was to just revive that all the diseases which we inherit from our parents are like diabetes or obesity are all due to misplaced epigenetic tags. So, and the mention, as in the video mentioned, the certain types of environmental factors also affect the epigenetic of a person. And we would be going through that in now. Epigenetics and environment, as we already know from the definition, are interrelated. Different environments have different effects over our epigenetics. Like if a mother, if we are taking a good, if we take good care of fetal in prenatal care or postnatal environment, it's good for a child. Then certainly it would develop a better. It would have different kind of epigenetic tags on their genes, like there are certain genes which control obesity. If you are given good nourishment during your early childhood or during your prenatal stage, then you won't be developing obesity due to genetics. But maybe in the later stages, it may be due to other factors. Second is pollution. Air pollution can pollution is a major factor which can change your epigenetics, especially air pollution. Several studies have shown that air pollution causes change in methyl tags on different genes. Our diet, as Arushi mentioned earlier, nutriepigenetic means that our diet also has certain role to play in our epigenetics. The lifestyle choices, how the hygienicity, uh, how the hygiene which we maintain, or the cleanliness which we are living in, that describes how our epigenetics or our genetics would look like. And especially the stresses also it also causes the change in our epigenetics. Now we'll be focusing on certain types of stresses. There are majorly four types of stresses are there: psychological or psychiatric, which develop due to uh, early early childhood mental abuse, or there could be induced stresses, or there could be physical stresses which may come due to certain kind of trauma, malnutrition, hyper exercise, or due to certain types of infections. And it may also be, in some cases, it may also be random. And now there have been several studies done over epigenetics on a special case, which is Chinese famine. Epigenetics in the cases of famine have already done in previous famines like Dutch famine and Netherlands famine, which occurred before during after World War II and before World War One. And all these studies show that epigenetics is high is linked has a great linkage with the diseases which we get, like especially like type 2 diabetes mellitus or obesity. In the Chinese famine case, the case study which I mentioned, IGF-2, which used IGF-2 to study the epigenetics of 188 participants, were divided into two groups: moderate or severe, which were based on the two different provinces which had different effects of famine, and they they used blood samples and extracted DNA from the white blood cells, and using bisulfite sequencing, they identified what uh, type of methylations are present on our genome. The finding was that there were in previous series they described seven different genes, out of which two I can name which are very important: IL-10 and IGF-2. IL-10 is responsible for the brain activity, and its absence can cause certain types of mental disorders like schizophrenia. What the entire entire study focused on the lipid profiling. It showed that uh, the people the child who have been in mothers in the gestation period of up to 6 months uh, during this Chinese famine had high cholesterol level, and had high lipid content, and they had obviously high levels of IGF-2 methylation. The gene knock, uh, since we cannot carry gene knockout series, which means removing a gene to study its effect, it showed that these studies were done in a mouse, which showed that if IGF-2 is removed from our genome, then it can cause nutritional deficiency, growth restriction, neuronal glycogen storage reduction, and several other things. The Chinese famine studies showed that there were high methylation on IGF-2. That means IGF-2 gene was completely suppressed, and therefore there were certain metabolic problems, like there were problems of cholesterol metabolism, and there were certain genes which were regulated by IGF-2, which is insulin growth factor 2, like which are involved in lipid metabolism also. And therefore this it showed that the people who were in people who were affected by the Chinese famine had high cholesterol level, and it had high low density lipoprotein cholesterol. Lipoproteins are another type of lipid which also get increased due to less metabolism of lipids. Before we go on further, I would stop here to for any questions. Any questions? I think no. I would like to call Sandhya so that she can initiate quiz.
Okay, hi. There's a link in the chat box, and you can access a quiz. Just a reminder that when you're, so it'll ask for your name. Just give in your full name that you used to register, because yeah, we have to keep track of who all have attempted the quiz. And yeah, it's a very simple quiz, so don't worry. Yeah, so just start joining. Okay, we need three more people. I've put the link again for anyone who might have missed it. Please join. Okay, we have 11 people, so I think everyone has joined. Um, I'll be starting the quiz. There are just about three questions to go. Okay, this is your last question. Okay, that was the end of the quiz. I hope you had fun answering that. Anyway, so as you know, this class we discussed the mechanics of epigenetic changes and how exactly it works. Um, from next class onwards, we're going to be focusing on how it's applied. Like, what's the point of trying? What's the point of studying epigenetic changes? What are the fields in which it's applied? Like how Arishi spoke about a few today, we'll be elaborating on quite a few of those to teach you the importance of the subject and what kind of a future it holds. Because it's a really emerging science. It's relatively recent, but it's been growing, and that clearly shows that how important it is. So we'll be focusing on that in the future sessions. For that, if any of you have any, we'll also be sending out this quiz as a Google form to those who might not be here. But if you've attempted the quiz right now, please don't attempt it again. But yeah, thank you. Over to Janvi now.
Hi everyone. Firstly, thank you so much for tuning in to the second session for epigenetics organized by Genesis, the Life Sciences Society, and Team KaryoUS. I'd like to thank our presenters here who have put in a lot of hard work to bring this edition for you guys. We really dug deep to break out the details of the mechanism that switch off and on the gene expression. We hope that you found this session useful and fun. Please do write to us and give us any feedback that you have, something that we can do at our end to meet your needs, any questions you want to be addressed, and we'll be happy to include them in the future sessions. We'll see you guys next week on the 8th of August at 6 p.m. The link for the same will be sent to you guys soon. Until then, again, feel free to reach out to us via email or on Instagram, and we hope you have a wonderful weekend ahead. Also, if we have two people, Science Tarography and Arzu, if you could please on the chat box put in the names using which you've registered so we can mark your attendance for this session. And otherwise, if there are no more questions, nothing else to address, we can call off the meeting. Okay, thank you so much, guys, and thanks for putting your names in the chat box. We'll see you next class.
Thank you.